friends. In this tutorial we'll be talking about the nucleic acids and it's about the nucleic acid metabolism. Nucleic acids are grouped into two different types, and actually if we break them down, they are mostly DNA and RNA, which are produced with the help of different bases which form nucleotides. So let's first talk about those nucleotides and how exactly those different types of nucleotides are produced inside our body. That's what we'll be looking. So these are the anabolic processes of producing nucleotides that actually give rise to nucleic acids of our body. Nucleotides are very important biochemical molecule. The unique portion of a nucleotide is the base that we know. The base can be of two different types: purine bases. Let me take a color here. Two types of bases that we see: purine bases and pyrimidine bases. Okay, and purines contain a double ring structure. You see the example as adenine, guanine. They contains double ring, while pyrimidines contain a single ring structure, like cytosine designated as C, thymine designated as T, and uracil designated as U. Very simple to remember. Now if you take one of them, which is different types of nucleoside will be formed when there is a sugar added to it, and then when there is multiple phosphates added to that sugar, it's converted into a nucleotide. So a base combined with ribose sugar simply make it nucleoside, and when the sugar is esterified with more phosphates to it, it's known as nucleotide. That's what we saw in the early animation. Now nucleic acids, nucleosides and nucleotides are needed for a number of different biological processes. They are used in RNA and DNA, as well as in 4 different types of coenzyme synthesis. For metabolic energy and derivatives are used in the signaling processes as well. And there are nucleobases, and those nucleobases that we talked about, adenine, thymine, guanine, cytosine, they can be synthesized in 2 different pathways, either de novo pathway or the salvaged pathway. The de novo pathway says that all these bases are produced from the scratch, okay, from the start, from the beginning, while the salvaged pathway states that there are fragments of nucleotides, fragments of bases and other materials already present all the time inside the cell. So they take any of those fragments and then utilize them to be converted into the desired base. So let's look at them. Okay, let's first talk about the pyrimidine synthesis process. Pyrimidine synthesis inside the cell can be mediated by 2 different pathways. One is the de novo pathway and another one is the salvaged pathway. The de novo pathway states the production of the pyrimidine from scratch, from the beginning of the synthesis. And the salvaged pathway dictates that process of existing molecules inside our cell and using them to produce the pyrimidine. Let's look at the process. First, the de novo process of the synthesis, then we'll look at the salvaged pathway later. This is the complete pathway list that is provided here, starting from this carbonate, and then it goes to like formation of many more intermediates throughout, and finally getting what we desire. That could be UTP or CTP or GTP or TTP or ATP, depends. Let's move on. The first step is the preparatory step, and in this case they produce Carbamoyl phosphate, okay, using bicarbonate and glutamine. Glutamine here act as a very important ingredient, and glutamine, when attached with bicarbonate, they use energy from ATP hydrolysis, and this step requires 2 molecules of ATP to be hydrolyzed to finally produce Carbamoyl phosphate. So as they use glutamine, they don't need ammonia to use, and that's a very good thing because ammonia can be toxic for the cell. Now once they produce this Carbamoyl phosphate, this enzyme that required to convert this carbonate and glutamine into the Carbamoyl phosphate is known as Carbamoyl phosphate synthase 2. Now this carbamoyl phosphate synthase 2 is under negative allosteric regulation by cytidine and uridine nucleotides. That means if you have cytidine or uridine nucleotides, in case of pyrimidine we only have the C and U, in purine we'll see A and G. Now if we have this UTP or CTP or UDP and CDP present in the cell, those things will negatively impact this first step of commitment, because this first step is irreversible in nature. Okay. Now carbamoyl phosphate 2 synthase 2 or synthase 2 is present in the cytosol, while carbamoyl phosphate synthase 1 is present in the mitochondria. And the carbamoyl phosphate synthase 1 that is present in mitochondria is used for the urea cycle, and that uses ammonia as the nitrogen source. Okay, but in cytosol we don't use that ammonia as the source in case of the first step. Instead of ammonia we use glutamine. So this is the step that is depicted here. We have the glutamine, we have the bicarbonate. The bicarbonate will be added to the glutamine, the structures, and they also need 2 molecules of ATP to be hydrolyzed for this process to take. And as a result what they will produce is a carbamoyl phosphate, where it has a nitrogen donor, that NH2 group. This group is not there directly from ammonia, it's from the glutamine, because glutamine carries this NH2 group. So this NH2 group is transferred to this COO, that bond there, to form a carbamoyl phosphate, and a phosphate group from the ATP hydrolysis, they'll drag this phosphate group and attach it to form the carbamoyl phosphate, and it converts the glutamine into glutamate or glutamic acid. So the glutamate can also be taken for other purposes inside the cell. So it's a very clever step to involve with. Now if we go to the second step, that is the step where the aspartate is added to make carbamoyl aspartate from carbamoyl phosphate. Aspartate is another amino acid, and you know, for the synthesis of pyrimidine and purine, you will see the involvement of a lot of amino acids, essential amino acids in our body. That's why those amino acids are very, very important as a source to generate the nucleotides. Now carbamoyl phosphate is added to the aspartate with the help of enzyme aspartate transcarbamylase. Now in this case they require this aspartate transcarbamylase enzyme to properly place the carbamoyl phosphate with the aspartate, and they'll form interstructure. Nothing is produced, nothing is released extra. They produce intertwined compound of carbamoyl aspartate and phosphonoacetyl L-aspartate, or PALA, phosphonoacetyl aspartate. So we call it phosphonoacetyl L-aspartate because it's the L form of that enzyme. It's also known as PALA or PALA. It's a very tight binding inhibitor of the aspartate transcarbamylase site. So use of PALA can prevent this whole process at this particular point. If we go to the next, after they produce carbamoyl aspartate, you see this is more of a open ring structure. Then rearrange this ring. Simply just imagine this ring to be rearranged. Just flip this base hydroxyl at that side and have a closed ring. Just simple flipping can give this structures very ease here. This flipping is actually making the closed ring, and once they form the closed ring of carbamoyl aspartate, they generate what is known as dihydroorotate. Okay, dihydroorotate is the same structure except for a water molecule released from the carbamoyl aspartate by converting into a circular form, a closed circular form. Okay, and the enzyme required for this process is dihydroorotase. That enzyme produces dihydroorotate. After that, the oxidation of the dihydroorotate is mediated to convert it into orotate. It involves an electron acceptor bound to the outside of the inner mitochondrial membrane, and that electron acceptor here is FAD. FAD is present in high amount in humans, which carries that electron, and FAD is reduced to FADH2 to convert and rearrange dihydroorotate to orotate. A simple rearrangement. And the enzyme required for that, as FAD and electron transfer is going on, we know always if FAD and NAD is linked with any enzymatic reaction, there is the enzyme dehydrogenase plays important role. And exactly same thing here. The enzyme involved in this process is known as dihydroorotate dehydrogenase enzyme. And the key point for this reaction is that the enzyme is present on outer surface of the inner mitochondrial membrane. And this electron acceptor is different in eukaryote and some bacteria. That makes a very important point, that makes this specific part of the pyrimidine synthesis as a potential target for the development of antibiotics. Let's move on to the next part. Now, sometimes they also need here, with the help of another specific intermediate that is known as phosphororibosyl 1-pyrophosphate or PRPP. But phosphororibosyl 1-pyrophosphate is not normally present. It should be present and mediated by converting ribose 5-phosphate into PRPP, and that is made because here this phosphororibosyl 1-pyrophosphate is under the negative allosteric regulation with the help of ADP and GDP, because the presence of ATP influence ribose 5-phosphate to be converted into PRPP, while presence of ADP and GDP prevent this conversion. Now this PRPP is also a precursor for the purine biosynthesis, and it is used in a number of different nucleotide salvage pathways as well. Now PRPP is also required to combine with orotate to produce OMP, which we'll study later in the next step. But in this case, you will see ribose 5-phosphate utilizes two different phosphate groups from ATP, converting ATP into AMP because it drags two phosphates out of ATP and attaches to the first carbon, and this is mostly like with the hydroxyl region, it attaches the two phosphate groups linked with each other. That's what is known as pyrophosphoryl 1-pyrophosphate, because two phosphates are transferred and attached to the ribose 5-phosphate sugar. Now the latest step where we combine PRPP to the orotate. This addition, it releases pyrophosphate. If you look at here, this is orotate, the ring, and this is PRPP, which we just saw forming. Now these two will combine with each other, and as a result of that, pyrophosphate will be released. And once pyrophosphate is released, the hydrolysis of pyrophosphate will act as an energy source to drive this process in the forward direction. Okay. Now, as the pyrophosphate is hydrolyzed and to drive the reaction forward, it makes the addition of this reaction biologically irreversible. So it cannot go back. It's irreversible in reaction, and the product is orthonidine monophosphate or OMP. Orotidine monophosphate. Now though in this picture it's shown like it can go backward, but it's not actually possible. It's irreversible in nature, so the arrow should not be kept like this here. Let's move on to the next part. Now once we produce this OMP, orotidine monophosphate, the same polypeptide chain that catalyzed the previous sugar transfer carries out the same type of transfer of converting OMP into UMP, and that's simply releasing of carbon dioxide from OMP. If you release the carbon dioxide from OMP, it converts it into UMP. You see the carbon dioxide linked with the with orotate here. That CO2 is simply released, and there's a closed bond from there, and then we get this UMP. Once we get UMP, rest of the situations become plenty of easier, because these nucleotides can easily be converted from UMP to UTP, UMP to UDP, from mono to di to triphosphates inside the cell using ATP as a phosphate donor, and it can achieve that any time with the help of enzyme known as Nucleoside Kinase, and it can simply transfers phosphate group from ATP to the desired base. Actually it's not base, but they transfer this to the sugar to convert that nucleotide into mono to di to tri and others. Okay. Now if you look at the nucleoside kinase, nucleoside diphosphate kinase reversibly transfer a high energy phosphate from a nucleoside triphosphate to a nucleoside diphosphate. It has broad substrate specificity and works for various nucleotides. While nucleoside monophosphate kinase accomplishes a similar task, it transfers two high energy phosphates from one nucleoside triphosphate to a nucleoside monophosphate. That's how the whole process is done. If we look at every process of this, imagine the TTP transfer the phosphate to ADP to convert it into ATP. Okay, so it's simply from tri to mono to convert the mono into di, it will remain. So that's how it transfers, simply interchange of the phosphate groups in between. Okay, for example, if we look at here, CTP transfer it to the ADP converting into ATP, and CTP becomes CDP. Vice versa, they can start transferring this process again and again to establish whatever type of conversion that they require. In the last part of this process, simply again the transfer of UTP to CTP, the conversion of U to C, and that is mediated by utilizing ATP in the middle, so provide the phosphate that will be tagged to uracil. And we know if we transfer a phosphate to uracil in a specific position, it will convert that U into C. And that's the conversion mediated. And again, for this process they require the nitrogen donor. That's also a very important job. And the nitrogen donor in this case also is glutamine, as it has the NH2 group extra. We don't use ammonia because ammonia is toxic to the cell. Now this is about the de novo pathway of the pyrimidine synthesis. If you look at the salvage pathway, most pyrimidines used by the body are not created by de novo pathway, because most are extracted from digested food or salvaged from cell breakdown products, because it's a better way to utilize and recycle the materials that a cell is producing over its lifespan. Here are some examples of the pyrimidine salvage reactions. Now we can see each of these reactions step by step. There is no other step by step reactions that will develop. There is also simple one or two different stages of chemical reactions that give rise to the target pyrimidine that we require. And the target sequences can be TMP, UMP and structures like that. If you go to the first one, we take a thymidine and we also have ATP. Now the thymidine had ATP, ATP transfers a phosphate to this and converts thymidine monophosphate, and ATP becomes ADP. This is a process which is catalyzed by thymidine kinase. The second one, say uridine, and we have a ATP that converts uridine to uridine monophosphate and AMP. So for the process of the salvaged pathway, we have ready availability of thymidine, uridine, deoxycytidine, thymidine and thymine and pyrimidine and so many other breakdown products inside the cell. So it's not building from a scratch, it's not building from zero, it's already there. It's simply replenish it, and little modifications are the maximum that we require. For example, for conversion of uridine into UMP, we require uridine kinase. For the conversion of deoxycytidine to DCMP, we need deoxycytidine kinase. So simple kinase enzymes can transfer phosphate group taking from ATP to the target recycling nucleotide factors, converting them into the desired pyrimidine and purine bases in all the cases. Now for example, thymine can also be paired with deoxyribose 1-phosphate. And once we pair thymine into deoxyribose 1-phosphate, it releases a phosphate group out and it combines that sugar with thymine to form thymidine, which is a nucleoside, and then it will be converted into nucleotide once we transfer more phosphate groups there. Now similarly, pyrimidine, if we tag with PRPP, it can also produce pyrimidine nucleoside 5' phosphate. So all these interchangeable chemical structures are formed, and those interchangeable structures can be modified by simple phosphorylation and dephosphorylation. If it requires the phosphorylation process to work, we have kinase to do the phosphorylation process. If it requires dephosphorylation or phosphatase activities, we have other phosphatase enzyme to bring that. And ultimately, by this fashion, we can recirculate between the different types of nucleotides inside the cell.